So it's really great to see that we've got a full room, nearly all the seats are taken. Welcome everybody. My name is Annika Lukeson, I'm Director of the Centre for Personalised Medicine. I'm really delighted to be co-hosting today's Stanley Ho Memorial Lecture together with the Oxford Martin School. It's a real privilege for us to be doing that. I'm going to hand over for the introductions to a recorded introduction from Mr. Ian Hewan, who is representing the Stanley Ho Memorial Foundation, on whose behalf this lecture is being held today.
Welcome to the Oxford Martin School and the Centre for Personalised Medicine's lecture by Professor Caroline Wright. This is the Dr. Stanley Ho Memorial Lecture. I am Ian Hewan, a trustee of the Dr. Stanley Ho Medical Development Foundation and member of the CPM's External Advisory Board. The foundation aims to enhance the quality of medical service and delivery in Macau, Hong Kong, and other regions of China by entering into academic partnerships with relevant worldwide organizations. It is our pleasure to have supported the Oxford Centre for Personalised Medicine since 2013. Dr. Stanley Ho passed away in Hong Kong at the age of 98 in May 2020. He was an extraordinary leader and a great philanthropist, and his legacy is his extensive altruistic efforts. The foundation honors Dr. Ho's memory by continuing his spirit of serving our communities and by supporting this annual memorial lecture. The Centre for Personalised Medicine is a communication, engagement, and research initiative based at the University of Oxford as a joint venture between the Centre for Human Genetics, part of the Nutfield Department of Medicine, and St. Anne's College. Its aim is to communicate on and engage with all aspects of personalized medicine with scientists, clinicians, policymakers, students, and the public. The Oxford Martin School brings together a vibrant community of scholars drawn from across the University of Oxford. Its research tackles issues of global significance with teams working towards real-world impact, from innovations in science, medicine, and technology, through to expert advice and policy recommendations. We are delighted that tonight's lecture is in collaboration with the Oxford Martin School. It gives me great pleasure to introduce tonight's speaker, Professor Caroline Wright, whose lecture is entitled Revisiting Genetic Determinism, Evidence from Large Population Cohorts. Professor Caroline Wright is a professor of genomic medicine in the Department of Clinical and Biomedical Sciences at the University of Exeter, UK, and academic director of the UK National Health Service Rare and Inherited Disease Genomic Network of Excellence. Her main research interests are in the clinical application of genome-wide assays for the diagnosis of rare diseases, and she has expertise in analyzing exome genome sequence data, particularly in rare pediatric disorders. She is researching novel genetic causes of disease, thereby improving the interpretation of rare genetic variants, understanding the penetrance of rare pathogenic variants, and exploring the policy and ethical issues associated with implementation of genome-wide sequencing in healthcare.
So I'm not going to add very much to that, apart from to say that Caroline is a real polymath, and as Ian's already said, she's going to be talking about the science today, but I've also worked with her over the last more than 15 years. I think we worked out on ethical issues, policy-making issues, and sometimes Caroline comes to Oxford for piano, to play the piano at very advanced levels. So she tells me she hasn't had enough time for that recently, but hopefully you'll be able to fit that in as well. So Caroline, over to you. Thank you very much. Thank you, and thank you for that introduction. And thank you for the opportunity to speak today. I've had a really wonderful day in Oxford with staff at the CPM discussing some of our collaborative work, and I'm really looking forward to further discussions around these issues. So I've got a slightly provocative title here, Revisiting Genetic Determinism. Obviously, that was partly to pique your interest. And genetic determinism is a concept that's been around for many decades. You can see in this cartoon, hopefully you can read at the back, a chap saying to a judge, "I'm not guilty by reason of genetic determinism, Your Honour." Now, of course, we know now that genetics is more complicated than this. We know that there's a number of factors that influence an individual's behaviour, their traits and their diseases. But nonetheless, we see the humour, and the idea still has traction, I think, in modern genetics. So what I want to do today is tell you a little bit about genetic determinism and really how I'm going to talk about it, which is through the lens of monogenic conditions, which I'm going to talk about in a little bit of detail. Then most of the lecture I'll spend talking about some of the work we have been doing in Exeter, looking at a phenomenon known as incomplete penetrance, and we've been using large population cohorts to look at this. And I'm going to take you through 3 particular case studies to illustrate this. A diagnostic study, incidental findings, which I'll introduce, and then finally screening, before I draw a few conclusions. So what do I mean by genetic determinism? This is a quote I've taken from a book, that genetic determinism is essentially the belief that an organism's phenotype is determined by their genotype alone, that a phenotype is essentially a necessary occurrence, that you can predict with accuracy how an individual will develop their phenotypes, their traits, diseases, behaviours, based on their genotype without looking at social and environmental factors. And as I said, we know this is a largely outdated concept in many areas. We know that behaviour is much more complicated than this. Many traits have multiple genetic and environmental components. Nonetheless, I think we still hear about genetic determinism quite regularly. It is still an assumed concept in much of the media coverage of some genetic findings, where where we hear about concepts such as genetic fate. And a recent review looking at genome testing in medicine said that some of the confusion around how we use genomic testing is based on misconceptions of genetic determinism. And this was said by Les Biesecker, who's chief of the Centre for Precision Health Research at the NIH. So I really wanted to try and understand, why is it that we're still talking about this? Why is it difficult? And how can we try to understand it better? So we know that our phenotypes, the traits, behaviours, diseases, everything we present is a combination of genotype and environment. And it's not just nature or nurture, it's nature and nurture. The two interact, they are not independent. Nonetheless, there are some traits and diseases that are more environmental and some that are more genetic. So that the size of the component can vary, as you can see on this spectrum. So at one end, the more environmental conditions, such as scurvy or tuberculosis, they do have a genetic component, but they are primarily environmental. As you move through the spectrum, you have a set of complex diseases, which we might describe as polygenic. They have a multiple genetic components, as well as environmental factors. And towards the other end, the red end, which we might think is primarily genetic, where the largest factor that's predictive is genetic. So conditions such as cystic fibrosis or muscular dystrophy. And these are what we might term monogenic conditions. So how do we think about these in the modern world of genomic medicine and our understanding of determinism? And what can we say using large population cohorts about monogenic conditions? So I thought we might need a little bit of background for those of you who are not primarily geneticists. So when I say the genome, I'm talking about the entire genetic code for an individual human. And we all have 6 billion base pairs in our cells. So an enormous amount of sequence information. And we all have around 5 million variants. So that's 5 million or so positions in your genome that's different from someone else's. That means that most of those variants are benign or harmless. They are part of an evolutionary diversity, but they don't relate to disease. We all have approximately 2 copies of about 20,000 protein coding genes.
That's absolutely normal. And the concept that there is a gene for any disease or trait, which you might read in the media, is absolute nonsense. We do not have a gene for anything. We have genes that code for proteins that do jobs. And of our 5 million or so variants, we all have hundreds that are linked with different diseases. And some of those might predispose to disease and increase your risk slightly, like some minor environmental factors. And some will actually cause very rare genetic diseases. And one of the things that we can use to understand the difference between those is looking at the frequency of those variants in the population. And by and large, it's true to say that common variants down here in the corner in brown, common genetic variants that are present in a large proportion of the population will have a relatively small effect size. They will not have a very large deleterious effect in the population because we've had evolution to get rid of really, really strongly deleterious variants. We can look at their effect on diseases and traits using genome-wide association studies, and many, many hundreds of variants, thousands now, have been linked with different diseases. As you move up the spectrum and you get rarer and rarer, so variants that are rare in the population, the chance that they can have a larger effect increases. So in the top box, highlighted in blue there, rare, extremely rare variants can cause Mendelian or monogenic conditions. And that's primarily what I'll be talking about today. And we've got thousands of variants along that spectrum, particularly in those two circles, and relatively few examples in the other circles, because we probably won't find common variants with big effect sizes, and it's still quite hard to find rare variants that have small effect sizes. But there are plenty of rare variants, many of which will have small effect sizes or be benign. They're just harder to look at because there's fewer of them in our data sets. So what do I mean by these Mendelian conditions? So monogenic or Mendelian conditions are those that are primarily caused by rare genetic variants in a single gene. And their inheritance patterns are fairly predictable. So we draw these sorts of family pedigrees where you can see an affected individual tracks through the family. You've got a 3-generation family here, male and female, represented by squares and circles. And that enables predictive testing through families. So we do often genetic testing for these sorts of conditions to enable prediction for future family members and also targeted treatment. And I think these conditions are largely assumed to be primarily deterministic. If you have the pathogenic variant that causes the disease, you are likely to develop, or your family will have that condition. And genetic determinism, I think, is still an assumption in a lot of monogenic conditions. The last thing to say about these monogenic conditions is there are thousands of them. Although they are individually rare, they are collectively quite common. There are some which are caused by variants in just a single gene, so cystic fibrosis, for example, or Huntington's disease, which are down the bottom here in this graph, where you have a very specific phenotype, a specific disease caused by variants in one gene. There are also monogenic conditions caused by many, many genes. So an individual still has one variant in one gene, but there are many similar diseases caused by variants in different genes. And as you go up the spectrum here, you can see up in intellectual disability, so monogenic forms of intellectual disability, there are hundreds and hundreds of genes known where deleterious, rare deleterious variants in these genes can cause those conditions. And there are rare monogenic subsets of many common diseases. And because of this immense heterogeneity, increasingly we use genome-wide sequencing, so sequencing the entire genetic code, in order to diagnose the cause of monogenic conditions, particularly things like intellectual disability. We can't possibly guess the individual gene based on the clinical appearance of an individual, and so we've started to sequence whole genomes. And that, of course, creates immense challenges for interpreting our variants. If we've got 5 million variants per genome, and maybe only one that's causing the disease, we'll come on to whether there's more than one in a moment, finding that needle in a haystack is a real challenge. So genome-wide sequencing in healthcare, there've been really two main areas of progress here, in my view. One is sequencing patients with rare conditions, where perhaps 80% of these rare conditions are caused by a monogenic condition, so a single genetic cause. As I say, they are individually rare, so fewer than 1 in 2,000 individuals, or perhaps much, much rarer than that. But if you add them all up, the thousands and thousands, they're quite a large public health burden, affecting perhaps 6% of the population. The other major thing that's happened over the last 10 or so years is an increasing sequencing of populations, so large common disease cohorts, but also control cohorts. Enormous numbers of individuals have now had their genome sequenced and their variants have been identified. That enables us to look at variant frequencies in different populations and think about their potential effect sizes. It allows us to see parts of the genome that vary a lot or are under evolutionary constraint. And in particular, it allows us to investigate disease-causing variants or disease-predisposing variants in larger and larger cohorts, so we can get at rarer and rarer variants and their implications for individuals and populations. So all of that as background, really, to the key question I want to ask in this lecture, which is, does a monogenic disease-causing genetic variant always cause disease? And if not, why not? So I'm going to take you through 3 case studies, particularly looking at this and their implications for how we use genome sequencing in the future. So the first one is perhaps the simplest to understand, is a diagnostic scenario. So I'm going to introduce you to the Smith family here, who are a family with 2 children, both of whom are affected by a severe syndromic developmental disorder. So this manifests by dysmorphic features, hearing loss, skeletal abnormalities, autism, and severe intellectual disability. And so their genomes are sequenced now through our Genomic Medicine service in the UK on the NHS to try to find the cause of this particular condition. What comes back from that test is that there's a genetic variant in a particular gene called ankyrin D11 that explains the 2 children's condition, absolutely explains it and is the diagnosis. But what's puzzling is that it's also carried by the unaffected father. Now, I should say, this is a fictitious example. All my case studies are fictitious, but there are individuals in a large study that I'm involved with, the Deciphering Developmental Disorder study, with exactly this scenario. Apparently unaffected fathers who have passed on what seems to be a fully penetrant pathogenic variant in this particular gene, ankyrin D11. So how is this possible? So this brings me on to the concept of incomplete penetrance. And penetrance is simply the probability that an individual with a pathogenic disease-causing variant develops the particular condition. So at the top here, in a number of individuals, you can see a completely penetrant phenotype, where every individual who carries that genetic change also has the condition. The next line down, we have incomplete penetrance. So there's 2 individuals there who carry the genetic variant and don't develop the condition. So across that population, it would be considered to be 60% penetrant. We also have a separate but related concept of expressivity. I'm sorry, that hasn't shown up quite right. Expressivity is the degree to which a trait expression differs quantitatively or qualitatively between individuals. So what these boxes should show is slightly different shades of blue, and that's what they're showing on my screen, but not yours, I apologize. So they should be going from dark blue through various shades of lighter blue. Everybody's affected, but some people are less severely affected than others. And similarly, if you had some different shades of blue here, you would see some people have fully penetrant severe condition, and a couple of individuals have a less severe condition. And some individuals are completely unaffected. So both concepts exist within monogenic conditions. So how can we try to understand what's going on here? And much of what we think about here is modifiers. So modifiers that can affect the penetrance of a particular genetic variant in a particular gene associated with a particular condition. I've just got a diagram here indicating a gene with its various protein coding parts here. It's expressed, so the gene is copied into RNA to make protein in particular cells. So one of the things that can be different in terms of penetrance is different causal variants. So different variants in the same gene can be associated with similar conditions or different conditions with a different level of penetrance, which we'll come back to. Penetrance can also be affected by other genetic variants. So, for example, genetic variants in these promoters or enhancer regions. These are parts of the genome that are non-coding, but they regulate the expression of genes. So variants in these parts of the gene could potentially turn up or down the expression of a gene. So you essentially have more protein product in a cell or less. And that can obviously affect penetrance. There's no doubt that random stochastic factors also play a role in penetrance. Very early on in development in particular, very small numbers of transcription factors in different cells can make the difference in a gradient as to how a cell develops. And that can just change with time. So we can have some randomness in whether particular genes are expressed, where and when and how. And then finally, of course, the environment can significantly modify penetrance. Even in monogenic conditions, there are certainly conditions that will only be present given a particular environmental modifier, so the presence of a particular food or drug, for example. And other exposures such as age, as we'll see, and sex, so other wider things can affect the penetrance. So can we use any of these to explain this particular case study? So the first one, there's absolutely no doubt in this case that this gene, ankyrin D11, this sort of variant in this sort of gene is very, very well proven to be associated with this type of condition, KBG syndrome, though it's normally caused by what are called de novo, or new mutations that have just occurred in that individual, so they're not inherited usually. The genetic variant in this particular case is definitively pathogenic. It causes a complete loss of function of this protein, one copy of this protein. And it's the same in the child, or the children, and the dad. So the different variant can't explain the difference. It is possible that the father only carries it in a small subset of his cells. So rather than being present in all the cells, it could be present in a smaller subset, and therefore he has it doesn't actually display any phenotype. That isn't the case here. He's got it in every cell. So the next question we're going to look at is maybe there are other genetic variants. Either the unaffected father carries additional genetic variants that compensate for that pathogenic variant, or the child carries, or the children in this case, carry additional variants that can exacerbate it. Obviously, the environment can have an effect, but we don't know of any environmental modifiers of KBG specifically, and there's a largely shared familial environment in this case. And there's also potential for random stochastic factors, which have been studied very nicely in things in models like C. elegans, and as you'll appreciate, much harder to study in humans. So what we've been able to really look at in these large population cohorts is the effect of other genetic variants on that primary causal variant. So what we're trying to do here is take a pathogenic variant that's been found in patients or families with monogenic condition and ask the question of, if we see that variant in healthy individuals who don't necessarily have any symptoms, what do we find? And for this, I'm going to draw very heavily on the wonderful UK Biobank. This is a really groundbreaking study that we have in the UK, a population cohort of about 500,000 volunteers, average age was about 56 at recruitment. And we have an enormous amount of data on these individuals. We have genome sequencing data, we have questionnaire data at recruitment, we have linked health data. These were not recruited as a disease cohort, these are primarily a population control. As a result of which, we see a lot of healthy volunteer bias and survival bias. So actually around 5% of individuals who were invited into the study said yes and joined it. And they are, by and large, they're sort of dying at a slower rate than the average UK population at the same age, and they are healthier and wealthier. They are a selected group of individuals. This means that they really should be depleted of severe monogenic conditions, particularly early onset developmental disorders, which certainly would reduce your ability to give, to have capacity to give consent for this sort of project. So they're a really interesting cohort to look at to see, do we even see these sorts of variants at all? And if so, what's their effect? Now, obviously, these variants are really rare. So it's fairly unusual to be able to look at individual variants. So we need to group similar variants of similar effect together to investigate their impact, which is what we've done. So if we look at developmental disorder, specifically in Biobank, we've grouped hundreds of genes like ankyrin D11 together where variants are known to cause monogenic developmental disorders. And what we find is thousands of individuals in UK Biobank carrying what appear to be rare, damaging variants. The sorts of variants, if you saw them in clinic, you would report them back as likely diagnostic. And we also see that they are associated with similar phenotypes that might indicate a milder or subclinical presentation in UK Biobank. So they are associated with a reduced fluid intelligence, a reduced memory, a shorter stature, fewer children, fewer years in education and fewer degrees, a lower chance of employment, and indeed a lower income. They're also associated with an increase in BMI, an increased chance that they're unable to work, an increased deprivation index, and indeed some of them have had diagnosis of a developmental disorder, and of course that's more likely with these individuals. And so we wanted to see what's going on here and what could be explaining this. And really the effect size we've seen can be modified by two particular things: the variant and its location, and indeed additional variants. So looking first at the variant and its location, so different variants, even in the same gene, but certainly across different genes, can have different effects. And we see that more deleterious variants have larger effects. So large deletions, for example, that might affect multiple genes have a much bigger effect than smaller variants. Those that are predicted to lose the function entirely of a gene have a bigger effect than those that just have a single amino acid change. However, there are specific examples where we can see a molecular rescue mechanism that we think might explain cases of incomplete penetrance. So just this one example of GATA6, which is associated with a congenital heart disease and pancreatic agenesis. I've grouped sets of variants here, blue on one side, which are seen in UK Biobank and population cohorts. They are predicted to truncate the protein, but they appear to have no phenotype. They do not seem to cause the disease. Whereas the ones in the red, which you can see cluster over here, have been seen in patients and really do have an effect that they truncate the protein and they destroy its function. And what we think is going on here is that the protein itself is being made from two alternative start positions along the RNA. And if you have the truncating variant early on, then there's a rescue mechanism, a molecular rescue mechanism, that means that that variant doesn't have an effect in that individual. We've also looked at additional variants and how they might alter penetrance. So there's a couple of different types of variants here. One is multiple large effect variants, so individuals might carry more than one of these rare deleterious variants. And we've also looked at the effect of multiple small effect common variants. So these are generally individually associated with particular traits. So developmentally relevant traits here might be educational attainment or cognitive ability, height, various things that I mentioned earlier. And an individual will carry a certain number of these common variants. There might be hundreds that have been associated with the trait. And we can essentially tally up the number of associated variants in one individual and work out what we call their polygenic score. So individuals who carry very few of these risk variants will have a low score, whilst individuals with multiple of the risk variants would have a higher score. And as you can see, the majority of the population will be in the middle of the curve here. And then we can ask the question, does this score, this polygenic score, affect the penetrance of the rare deleterious variants, that primary rare variant? So it could compensate or it could exacerbate the effect. And indeed, that's exactly what we see in developmental disorder. So we see that rare and common variants actually have an additive effect. So I've plotted here the polygenic score in Biobank for educational attainment, but we see similar things for a number of other traits we've looked at, and the effect on fluid intelligence on the y-axis. And you can see that there's, as you would expect, a line where a lower score for educational attainment is associated with a lower intelligence and a higher score with a higher one, as you would expect. What's interesting is when you pile on top of that individuals who carry these rare deleterious variants in developmental disorder genes, you've got 1, 2, and more than 3. So one individual carrying multiple variants. And you can see that each time the score drops a little bit. However, even those who've got 3 or more pathogenic variants are substantially better off if they're at this end of the curve than at that end of the curve. So their polygenic score is additive on top of their rare variant score. So these variants are acting additively. So what are the implications here for the Smith family? We don't have an absolute answer. We don't know the exact modifier in this case. One of the problems is it's hard to find a final diagnosis in this case. Sometimes these sorts of diagnoses will be delayed because we would exclude this kind of variant in our initial look at genome data, assuming it to be benign if it's inherited. It makes it difficult to counsel the family about what the future risk to other relatives might be. And of course, there are additional modifiers we don't know about. There's probably a high recurrence risk, of course, in this family. And some reverse phenotyping often occurs. So clinicians will go back and we'll discover actually perhaps the father was slightly affected when they were younger. That varies, and sometimes they are affected and sometimes they're not. But sometimes we can end up going from the genotype to the phenotype. In this scenario, that makes some sense. I'm going to change tack a little bit now and move into a slightly different scenario. So my last two scenarios actually are not diagnostic. They are where we find a genotype in someone who doesn't have symptoms of that particular condition in their family. So the first one is an incidental finding, so a finding that's essentially accidentally discovered that's not related to the reason for sequencing. So I'll introduce you to Diane, a 50-year-old healthy woman who's had her genome sequenced, similarly because she has a child with a developmental disorder, nothing to do with her. And she's found to carry a genetic variant in the BRCA1 gene that's linked with very high risk of early onset breast and ovarian cancer. She has no family history of cancer and no symptoms of cancer. What should she do? Now, variants in these BRCA genes, tumor suppressors, and they're thought to account for around 5% of breast cancers in the UK. And she's heard about BRCA, she's read about it in the news, she has friends with breast cancer. This is a pretty frightening piece of information. And she's got various options in front of her as to what she can do. So again, we're going to look here, what can we learn from population cohorts about how we should interpret these sorts of variants found incidentally when someone doesn't have a family history or symptoms of the condition. And here what we have to remember is the background, the prior probability that she has breast cancer is relatively low. That's not why she was tested, and it's unlikely she has necessarily a family history. So BRCA is a little bit different from the Anchorin D11 example, and we already know that it displays incomplete penetrance and age-dependent penetrance. So this is a graph of the risk of breast cancer increasing, as you can see, with age. And this comes from a meta-analysis of a number of different studies. But the risk for BRCA1 carriers across this meta-analysis is around 60% at age 60 years. And we know there are various modifiers. For example, the polygenic score can affect this by about 10% or more. And we know that family history affects it, so multiple affected family members will increase your risk of breast cancer. What we were interested to see is what if you looked in unselected population cohorts, because most of this meta-analysis comes from family studies or disease cohorts where individuals already have the condition or indeed are enriched for BRCA carriers. And what we see in UK Biobank is around half the level of penetrance. So in this unselected group, the penetrance is more around 30% to the age of 60. And that's primarily because there are different modifiers. Whatever the modifiers may be that we don't know about, they might be enriched in a family that's got multiple affected members as well as that highly penetrant variant, but they may be absent in a population cohort where there's no family history. And the other thing we looked at was to see how does family history affect that in this background cohort, because of course many people do have a family history of breast cancer. So this, again, is a survival curve for how long versus age, and this is women, are free of breast cancer. And the background population is just shown there in blue. And if I just add on top those who've got a family history of breast cancer, but no predisposing variant like a BRCA1 variant. And you can see that's a little bit more likely to get breast cancer, and they get a little bit earlier. The next line down is in green, and these are individuals who carry a BRCA1 pathogenic variant, but they have no family history. And the furthest down in the yellow is those that have a BRCA1 variant, and they also have a family history. And you can see that it has quite a big effect on modifying the penetrance. And actually that 30% figure really splits into about 44% for those with a family history, and only around 23% for those without. So a very big difference versus what we've seen in clinical cohorts. And this obviously has pretty major implications for how Diane is counselled. The risk estimates are much lower if these variants are found incidentally, and this informs decisions about whether she has regular screening or whether she has prophylactic treatment. And there's quite a big difference between pathogenic variant found incidentally, which may only be slightly above the population risk, versus those found in families with symptoms. There are some caveats to this, of course. I said that we have a healthy volunteer bias in UK Biobank. That means we have a bias towards lower penetrance variants and a depletion of the sorts of factors that might increase the penetrance. And at the other end, we have a bias in the affected families towards higher penetrance and an increase in the factors that increase their penetrance. So probably we have an upper and a lower bound of the risk estimate that's useful to help Diane make a decision. But I think the important thing here is that she knows she's different from Angelina Jolie, for example, who famously had a variant in this particular gene, who had an affected mother, affected auntie, multiple affected relatives. She's different from her friend who's just been diagnosed with breast cancer or who's found a lump. She doesn't have any of those background points, and so probably a better estimate is to think about this incidental rate. And then I'll jump into screening. So screening's a whole other kettle of fish again. So here we're looking at clinically unselected populations to see if we can find individuals who are at high risk of disease and target them for some sort of treatment to prevent disease early. So much like the incidental finding case, there's a much lower probability of an individual actually having the condition because they don't necessarily have any symptoms associated with it. And there's all sorts of screening available, and there's all sorts of consumer testing available for the worried well. I'm going to focus on one particular scenario. So baby Thomas here, born healthy and developing normally. And he has the standard newborn screening test, which is a heel prick test at 5 days, which tests for around 9 conditions in the UK, which are all preventable if treated very early. But he's also taking part in a research study into newborn screening. Now, there are quite a few of these studies being set up around the world, including one in the UK, to try to understand whether and how we might use genome sequencing as part of screening. He's found to carry a genetic variant in a gene linked with early onset monogenic form of diabetes. So what happens next, and what data can we use to understand what's going on here? So why would you look at monogenic diabetes? There are, obviously diabetes is not a rare disease, but there are monogenic rare subtypes that are caused by individual variants and individual genes. And they're important to diagnose early because they have major implications for treatment. Some occur in the neonatal period, some in early childhood. And rather than being treated with insulin as a standard for many, they're treated with sulfonylurea, which can be given as a pill rather than an injection. It makes quite a big difference. So that's why we might look. But we need some evidence from the population cohorts to tell us what the expected penetrance is and how likely it is that baby Thomas is actually going to develop diabetes and therefore be relevant to tell him about this or tell his parents about this variant. So in this case, we've got a couple of different cohorts we've looked in. So we've got, on the pathogenic side, in the orange, we've got both patients who have been referred for monogenic diabetes testing, and I'm lucky at the University of Exeter we run an international service for testing individuals who are likely to have monogenic diabetes. So we have those who are diagnosed and their family members. And in the blue side here, the population cohorts, we've looked in 2 cohorts in this case, UK Biobank, but also a Geisinger Medical Center, which is a US hospital. And each of these cohorts has quite different background rates of diabetes. So obviously the patients, 100% have diabetes. Their families, it's around 60%. In the Geisinger Center, it's about 24%. And in the UK Biobank, it's about 6%. So very different background rates of diabetes and indeed causal variants within them. But we can look in each of these cohorts for these specific pathogenic variants to see, is this causing disease or not? And here we see a slightly mixed picture. So one of the genes, KCNJ11, it causes a neonatal onset diabetes. And in this particular gene, there's a relatively small number of very specific variants that cause a gain of function. So this gene actually is more active as a result of these variants. And they're largely absent from the population cohorts. There aren't any in UK Biobank. There aren't any in the Geisinger cohort. So we don't know much about their penetrance. We can't say anything, except that at least it is plausible that they have high penetrance. A couple of other genes are in a slightly different category, so HNF1 alpha and 4 alpha. These genes, there are numerous what are called loss of function variants in these genes, so those that reduce the function of these proteins, and those are present in population control. So we can look, in these cases, we can group the variants together and look to see are individuals developing diabetes, and does that change depending on our ascertainment. So I'll just talk you through this graph. So here we've got the proportion who are diabetes-free on the y-axis versus age. So the green here is the patients. So by definition, 100% of the patients in our cohort have got diabetes. That's how they were recruited, and they have a pathogenic variant in this HNF1 alpha. If you look at the black line next, these are their family members. They carry the variant, but many of them do not yet have diabetes, and they develop it slightly later. But still, the majority, as you can see, eventually do get diabetes. If we now jump to the hospital cohort, so these have gone to hospital for various reasons, which may or may not have been diabetes related, and you can see a slightly reduced, again, penetrance. If we looked at the age of 40, for example, perhaps only 50% of them who carry pathogenic variants in HNF1 alpha have developed diabetes. And then you'll not be surprised at all to learn that the blue line is UK Biobank, our population control here, which again shows a slightly lower penetrance. The age of onset is later again, which obviously has implications for whether we should be detecting this at the newborn stage. And many fewer individuals ultimately develop diabetes. And these genes are quite interesting because although often variants are extremely rare, and so we have to group them together, one of the variants in HNF4 alpha, which is a missense change, actually is common enough that we can look at the penetrance of just that one variant across these different cohorts. And for just that one variant, we see exactly the same pattern, the movement of a decrease in penetrance as we move away from clinical ascertainment. And if you're now ascertained in the population, it's hard to say that this is really a pathogenic variant at all when ascertained in the population. Perhaps it does increase the risk of diabetes, but it's clearly not having the same effect as it does in the patient cohort. So the implications here for baby Thomas really depend upon the gene and the variant and what action you can take. And the challenge for screening, of course, is if the variant is penetrant, perhaps there's monitoring or treatment that can be done to prevent disease or reduce the severity. But if the variant is not penetrant, this introduces an uncertainty as to how we manage what was otherwise a healthy newborn, and potentially unnecessary and harmful follow-up testing or treatment, which is something we really need to balance as a society. For an individual, a variant is penetrant or not, but across a population, we really need to think about how we balance the benefits and harms of screening individuals who don't have any symptoms of a particular disease, because it's really quite unlike diagnosis. And although we sometimes think of genomics as being different from other biomarkers, actually it's no exception, and it isn't deterministic in these cohorts. So really, my conclusion from these case studies is that incomplete penetrance and variable expressivity of monogenic disease-causing variants is not universal, but it is really widespread. So in the diagnostic scenario in the Smith family, we saw that you can see incomplete penetrance in monogenic affected families, which can really make it difficult to classify a variant and find a diagnosis. We see it in incidental findings, Diane's example, where a lower penetrance is very likely because there's a lower probability of disease, and so the impact of the variant is much harder to predict because there's no family history or symptoms. And then that's amplified even further in the genomic screening context, where we are specifically selecting individuals who don't necessarily have any symptoms of the disease at the point at which they're being sequenced. And so there's great potential for overdiagnosis here. And although I focused on one scenario, actually, this is a much wider issue. We have opportunistic screening that is mandated in diagnostic laboratories in the USA, looking for variants across more than 70 genes to report incidentally or secondary findings. There are multiple direct-to-consumer companies that will offer these sorts of tests for people who are interested. And really, we need to think about how to communicate the fact that this is really not deterministic information. If it's not, or even in a diagnostic context, it might not be, and it certainly isn't outside of that context. So clearly, ascertainment bias affects penetrance. So at the bottom end, we have a sort of genotype-first approach, where we might genotype a population and look at the penetrance, which we can see is much, much lower. And at the top end, we have clinical cohorts, where a phenotype-first approach is taken. Individuals present with a family history or set of conditions, and then we target our test or our analysis to that specific question. Probably these are upper and lower bounds because of the sorts of biases I've talked about. And the general population is somewhere in between. And what we need is more studies looking at more and more individuals ascertained in different ways, and particularly increasingly ethnically diverse, because this also will vary with ethnicity, to try and understand what's the most appropriate penetrance and risk estimate for an individual, dependent upon how their genetic variant was discovered. So what's the explanation for the differences between these studies? I felt it would be remiss of me not to mention that one of the explanations between different studies can be errors. Scientists are not perfect, tests are not perfect, Sometimes the genotypes can be wrong. And particularly when you group variants, it can be easy to overlook really good genotype quality. Gene-disease associations, it seems like a established fact that will remain true all the time, but actually gene-disease associations can be refuted based on new evidence. So some older publications, in particular, that predated these population cohorts, may have erroneous gene-disease associations. Similarly, the variant consequences might be wrong, so when we group variants together, they may or may not be appropriate for the disease of interest. So, for example, if your disease is caused by a gain of function, there's very little point looking at the penetrance of variants that cause a loss of function. They will not cause that disease. So quite a lot of curation, careful curation is needed for these sorts of studies. And of course, disease definition can make a big difference. In a clinical cohort, a disease may be very, very well and carefully defined with very careful phenotyping. In a population cohort, you are dependent upon what information you've asked or what information you can glean from GP or hospital records. So all of these can influence differences in these different studies. And I should say that they can be wrong in clinical studies or wrong in population studies or right in either. None of us are necessarily immune to error. Assuming we all did our job right and that errors are not, or errors at least are minimized, there are still molecular mechanisms that explain this non-penetrance or incomplete penetrance. I've talked about different genetic variants and biological rescue, and I've talked about modifiers, in particular additional variants, whether they're common small effect variants or multiple large effect rare variants, as well as the environment. And increasingly, we're thinking about this in terms of a liability threshold model of disease, where numerous factors may contribute to the development of a disease in an individual. And so some variants in some variants will be 100% penetrant in pretty much every case. If you see the variant, it will cause disease because the variant, is large enough that it will always push an individual above the clinical threshold for presentation. And the environment and additional modifiers won't change that. However, in the incompletely penetrant conditions, of which I think we are finding more and more monogenic diseases fit into this category, one large effect variant isn't sufficient by itself to cause disease, and there are additional factors. So, for example, a second large effect variant, so two large effect variants that interact. It could be a heavy polygenic load, it could be the environment, all of which combine to push an individual above their clinical threshold, whilst other individuals have the same large effect variant, but they lack those additional factors to push them above that threshold. And of course, we don't really know what these modifiers are, by and large. We know a little bit about some of them. We know that the polygenic score affects penetrance. We've seen that across multiple monogenic conditions. And that's a relatively easy thing to measure, and we have polygenic scores for many hundreds of diseases now. It's very likely that additional rare variants have an effect, but often they're very difficult to find. And so we know they're enriched in patients and families, but we don't know how to look for them in populations, which makes it really, really hard to interpret this individual rare large effect variant when you don't have a phenotype and you don't have a family history. So there's a sort of final thoughts, just reflecting back on that original quote. So some rare monogenic diseases are near deterministic. I think that we haven't thrown away monogenic disease and the link between a causal variant and a disease. But not all variants are fully penetrant. And in particular, the interpretation of a particular variant really depends on the context. So if that variant is ascertained through an individual who has been tested as a result of clinical symptoms, it's likely that the penetrance of the variant will be much higher than if they were ascertained through a population biobank, for example, or screening or some sort of incidental finding. And so we really need to be quite careful when we're predicting disease in individuals who don't yet show any symptoms and don't have a family history. And this is pretty standard, I think, in public health, thinking about how we think about screening. And the key thing here is that genetic is no exception, and genetic determinism is not going to be the rule. It's going to be unusual in these population settings. So with that, I will finish and just acknowledge some of the fantastic team in Exeter who I've worked with on much of this study, particularly Mike Whedon, a professor of bioinformatics, who's done a lot of the bioinformatic work, and Kash Patel, who's led much of the diabetes work with Andrew Hattersley, and also a couple of individuals at the bottom there, not in Exeter, Helen Firth, Matt Hulls, and Ron Zimmern, who've really helped shape my thinking about these sorts of issues over the years. Just to acknowledge also the MRC, who provided funding for the majority of these studies, and I'll stop there. Thank you for your attention, and I'm happy to take questions.